thing is over it.
Yeah, but like this is not supposed to be like taking it. It's like 45 minutes. So whenever you feel like, or
Don't have information for that. People probably keep rippling in, but that's how we should go. All right, everybody, we're going to go ahead and get started with today's seminar.
all right, well, thank you so much. Well, before I start, I would really wish to do this before, just hit a recording button, but, just for your reference, this was meant to be a job talk practice. And that's why it looks, probably a bit snobbish, pretentious, what have you. But the point is, I did the job talk before actually doing this. So, it's like, it kind of didn't serve the purpose, but here I am anyway, and I hope, you guys still could enjoy this. Yeah, so my name is Hengxing. I am an IGCB postdoc just, around the corner at CEAS. And today I'm going to talk about a bird's eye view of ecological communities under global change. Oh, hold on. Okay, so let's start with this photo by the Japanese photographer Sugimoto Hiroshi. So the backstory of this photo is that he asked himself, what do you get if you shoot a movie in a single frame? And the answer is that you get this shining blank screen with no image from the movie itself. And this is because the fundamental dimensions of the movies are lost, and those are space and time. And ecological dynamics in nature are just like movies. They unfold through space and time, and how species interact with each other can be very different across habitats and over months in a single year. But unfortunately, many ecological studies still only focus on either an overview of either dimensions or a snapshot at a certain space or at a certain time. And these generalizations may lose many details of how ecological dynamics unfold. And a static or generalized view of these ecological communities become really limiting when we are facing rapid changes in the global environment. So for example, we're all familiar with global warming, and climate change has also caused many extreme weather events as well. So this is a record-breaking heat dome event happened in the Pacific Northwest in 2021. And on the other hand, human activities such as agriculture and urbanization are also changing the landscape across the entire world. So as ecologists, the question we want to answer here is, how can we understand the response of biodiversity under these rapid global changes? So to answer this question, I would like to focus on one single taxon, the birds. So with over 10,000 species around the world, birds are the most diverse tetrapod on Earth. They're also providing essential ecosystem services, such as nutrient cycling, seed dispersal, and pest control. They also have very high cultural values, as bird watching are becoming increasingly popular around the world, and many times often supporting local economies. And because of these, birds are of very high conservation priorities. And they're also very mobile and dynamic, which make them ideal to study the responses of global change. And as many other groups, birds are not coping very well with global change. So I'm sure many of you have seen birds making the news a few years back when we realized that North America has lost about 3 billion birds since the past half century. And it is even scarier when you consider that means that 1 out of 4 birds are gone since 1970. But obviously, birds can't just fly away from all these problems, especially when the problem is so pressing and universal as global change. So what do? We have seen morphological changes among individuals of the same species. So studies have found that individuals in the single species have been shrinking their body sizes, but their appendages such as beak and wing are getting longer. And some hypothesize that these are responses to a warming climate because all of these help with heat dissipation. And birds are also shifting where they're living. So around the world, we have seen birds shifting their habitats upward on an elevational gradient like those blue arrows on the left, and also northward on latitudinal gradients like those blue arrows on the right. And these are, again, consistent with a warming climate because birds are tracking their suitable habitats when their old habitats become too hot to live. And birds are also changing when they're doing things, such as migration and breeding, the things we call phenology. Many species are migrating and breeding earlier in the season, as you can see with those declining lines on the left panel. And on the right panel, you can see that these earlier phenology, which are marked by those red shades, are pretty consistent across space. And these phenological shifts are, again, possibly because the warming is changing the timing of birds' essential resources, such as plants and insects. But there's one big issue. All these evidence are on the species level. As we know, species interact with each other all the time, and birds are constantly fighting each other, they are eating each other, and they're also helping each other. Thank you. So to answer this question of how we can understand the response of biodiversity on rapid global change, it is really important for us to consider the community aspect, focusing on two specific, well, facets, if you will. The first is the changes in community composition, and second is changes in species interactions. And through my research, I aim to use big ecological data and quantitative tools to shed light on the two aspects on the finer spatial and temporal scales. So let's start with the spatial dimension first. So suppose we have two species that are overlapping with each other spatially. If we sample at the site where the two species range overlap, you might get this mix of species and they're interacting with each other. Now, what happens if one species is shifting its range away from the other species? At the same site, you might see that this plot might have fewer individuals of that species, and that's going to change how many of these species are interacting with each other. And the same thing with two species with overlapping phenology, which basically just means that they co-occur at certain times in a year. So if one species shifts its phenology earlier, then at this point, it will become more abundant at the same time. And that will, again, change both the community composition and the species interactions. So apparently, both the spatial and temporal shifts in the species ranges can affect or profoundly impact community dynamics. So during grad school, I've explored how phenological shifts can affect community dynamics using theoretical models and experiments. And this is where the name dropping starts, so bear with me a bit. So I showed that the phenological shifts can affect coexistence of two species in local communities. And then I scaled it up to spatial communities, showing that phenological shifts can affect the biodiversity patterns in fragmented landscapes. And it also explored how timing can affect species interactions with more than two species involved, or those higher order interactions. And I also conducted experiments with specific systems such as competing insects and plant microfeedback. And with all these knowledge, I proposed a unified framework to understand the impact of timing on species interactions. But we're going to talk about something new today. So I've been focusing on the spatial dimension of range shifts. And today I'm going to talk about two new and exciting stories about how spatial range shifts in North American birds have been affecting the community composition via changing functional diversity, and also species interactions such as predator-prey interactions. And finally, I'll introduce a novel method to collect large amount of data on species interactions to better understand how they're affected by global change, which paves the road to connect both spatial and temporal dimensions. Okay, so let's start with the first story on functional diversity. And the key question to answer here is, how do spatial range shifts affect the functional composition of local communities? And this is a collaborative effort with Adam Smith from Carleton University in Canada, Yilan Song, a postdoc here at the University of Michigan, and my two co-advisors for my IGCB program, Kai and Brian. And this is also on bioarchive. Okay, so to set up this question, we'll have to go back to the point where I first mentioned the birds are changing their morphology over time. The key idea behind these actually go back all the way to the 19th century, when people first realized that birds living in higher and lower latitudes are kind of morphologically different. And specifically, to improve thermoregulation, birds in warmer climates should have smaller bodies, which increase their surface to volume ratio, and also larger appendages, which also help with the heat dissipation. Now, as climate warms, we might expect to see the same pattern, but over time. So to improve a thermoregulation in a warming climate, we should see birds getting smaller body sizes and larger appendages, and as a consequence, larger relative appendage sizes. Now, why is that important? So let's take an example of body mass. Among individuals of a single species, if the smaller body mass is indeed favored or adaptive under a warming climate, then we should see the more individuals of the species getting smaller, and the mean body mass of the species will get smaller, which is actually what we see for many species around the world. But how about on the community level? So following the same logic, if smaller body mass is indeed favored, then we should see the smaller body species also being favored. And the increase in their abundance will drive the mean body mass of the community down, if that makes sense. But is this necessarily true? Or in other words, does these species level trends in traits really predict the community level functional shifts in the same traits? So to answer this question, we need several pieces of data. First is the community composition. And for this, we use the North American breeding bird survey. And then we need some values of the functional traits. And with these data, we can calculate these community-weighted means for all the local communities over past 50 years. And for local community, will be defined as a community within 1 degree latitude longitudinal grids. And then we can associate all of these community-weighted means with the environmental data, and then to see basically how they're affecting or associating with these community-weighted mean or functional shifts. So I'm going to walk through each of these pieces in detail, starting with the community composition. North American Breeding Bird Survey is a highly structured survey run by the USGS and the Canadian Wildlife Services, with yearly surveys in the US and Canada since 1966. It has a very consistent protocol and very high spatial temporal coverages, making it ideal for our purposes. So to start, we first got the abundance of 470 species by fitting hierarchical Bayesian models with spatial autocorrelation on the raw data. So here's what it looks like. And this is black-bellied wisdom duck, extremely cute, in 1 grid in southern Texas. And here you can see those gray dots are the actual counts from the survey. And the blue line is the fitted abundance over time. And then we adjusted these fitted abundances based on the detectability of each species in the field. So for example, if you're counting songbirds, then you might undercount them because only the males sing. So you might have to time that abundance by 2. And then stacking abundance of each species over time in each local community, we eventually would get the community composition for more than 1,000 grids over 50 years in North America. And then we compiled key morphological and demographic traits from several sources. For morphological traits, we used where we aggregated beak and wing measurements using PCA. They used their first principal components as a holistic measurement of their sizes. We also get body mass and relative beak and wing length, which are simply just the log ratio of those length with body mass. And we also included some demographic traits that are essential for bird life history strategies, including clutch size, which is the number of eggs in each clutch, and also generation length, which is the age at the first reproduction. So let me explain a bit more on this mysterious community-weighted mean measurements. So that is basically just the average trait value of all species in the community weighted by the species abundance. So suppose that we have 2 species here in this community, and their relative abundances are A1 and A2, and their trait value or body mass are M1 and M2. So in this case, the community mean of body mass is simply just the relative abundance of species 1 times its body mass, plus the relative abundance of species 2 times its body mass. And now, why are we using this metric? First, it is very easy to interpret because these units are all the same. So the community mean of body mass is just in grams, which is the same as the body mass trait values. And second, it also summarizes the entire community very efficiently. And also, it is less affected by rare species because all of these are weighted by their relative abundances. So finally, let's move on to the environmental variables. We got key bioclimatic variables from the World Climate data set, which includes the mean temperature and precipitation in warmest quarters, and temperature seasonality and precipitation seasonalities. And the reason why we use warmest quarter is because that is when the North American birds are breeding usually, and that's also when these surveys were conducted. And because we know that habitat quality can greatly affect the fitness of individual bird species, we also included these human-induced land use changes, which include the proportions of agricultural land, the inhabited land, which simply means the land with human presence, and then settlements, which means dense and urban suburban areas, and also the wild land. So for each of these environmental variables, we've averaged them within each grid for over 50 years. And then we fit linear mixed effect models between the community means and these environmental variables. Now, let's go back to our expectation on a species level. that a warming climate should lead to smaller body masses, larger appendages, and as a consequence, larger relative appendage sizes. Now, do these species level expectations translate or hold onto the community level? Let's take a look at how the community weighted means of these 5 traits are associating with temperature. Now, starting off with mass, we can already see that there's a pretty positive association between temperature, which is quite unexpected from the species level studies that says the body mass should be decreasing with warmer temperature. And beak and wing sizes are positively associated with temperature, which are expected. But then the relative beak and wing length are, again, negatively associated with temperature, which are unexpected. So overall, we're seeing that these species level expectations don't really do a good job in predicting the community level functional shifts. So how can we explain this? One possibility is that maybe some other environmental factors are driving these functional shifts, which is sort of what we see here. So this is plotting associations between the community weighted mean of body mass with temperature and other human-induced land use change variables. And you can see that some of them are actually on the same level or at the same strength as the association with temperature. And looking across all the other traits, we see that sometimes these associations with human-induced land use change variables are sometimes even stronger than those with temperature. So what that means is that these human-induced land use change variables are really important in driving the community-weighted means of these traits. Now, we know that species-level trends in traits often do poorly in predicting community-level functional shifts. The second question is, who are the culprits, or what species have been causing these functional shifts over time? Now, before we answer this question, let's talk about 2 possible scenarios in which community weighted means could be changed. So for example, we have seen increasing community weighted mean of body mass over time, and this can be caused either by a modest increase in many large body species in the community, or by like the extreme increase of only a few large body species, whereas all the other species are remaining more or less the same. So one easy way of looking at this is to calculate species contributions to those temporal changes. And now how do we do that? There's some math involved. Let's start with writing out this equation of the temporal slope of community weighted mean for one trait, which is the covariance of that trait value over time and time over the variance of time. And then we remember that community weighted mean is simply the sum of all the relative abundances of each species times their own trait values. So plugging this back into that equation and with some math, and that I will not dive into, we get this very convenient conclusion that each species' contribution to those slopes are simply their abundance slope over time times its own trait values. Now that we know how to calculate these contributions, we can start with an example with this grid in northeastern Utah. And here we are decomposing the community weighted mean of body mass, or its temporal trend. So here we're looking at this histogram of these abundance or absolute values of contribution for each species. And we take the absolute value here because they can be either positive or negative. And looking at this, we can clearly see it is quite a skewed distribution, with about 10% of all species on the right-hand side of this line contributing greatly to these temporal trends, whereas the majority of the other species on the left-hand side of this line are basically doing very little. We can then scale this up to the entire North America by counting the number of local communities in which a species is among top 10% that contribute to these temporal trends of each trait. And then we can pool the top contributors for all those traits. And we are really surprised to see that only 25 out of those 470 species in our study are driving the functional shifts in North America. Now, the number might be surprising, but when we look at who they are, it probably could make a bit more sense. Even if you are not a bird person, you could possibly tell some of these names from the list, such as this thing. It's been really growing and taking over the entire universe over the past 50 years. It could be in your neighbor's lawns, it could be in the urban green areas, and there might be some walking in this building. It's been really taken off. But you also see something really interesting like ringneck pheasants, which are introduced species and have been really booming in the past 50 years in the Midwestern and Western agricultural fields. And on the flip side of things, you also see something declining. You see northern bobwhites declining precipitously over the past 50 years due to the degradation of their habitats, which are mostly pristine grasslands and prairies, and also common grackles due to the degradation of their wetland habitats. And I should also mention that these amazing illustrations are done by Olivia sitting right here. I did not know you were coming, but just so you know, I'm acknowledging you like across the board, regardless of whether you're coming or not. Yeah, and you'll see her name again later in this talk. Okay, so far, we have identified these top 25 species that have been driven functional shifts in North America, and they're also greatly influenced by human activities. Now, the third question we have is how well can these top contributors explain these functional shifts that we have expected or what we have observed? So here's a recap of these associations between the community mean of 5 traits with temperature, with those highlighted traits meaning that they are unexpected. We then divide the complete community into 2 parts, one with the top contributors only, which we call top, and the other one with all the rest of the species, which we call standard. And we fit the same, we calculate community mean for these subcommunities separately, and then also fit the same the linear mixed effects models. Now, comparing among the 3 communities, we can clearly see that for traits that are showing the unexpected associations, the body mass, the relative beak length, and relative wing length, the top contributors are further up or down in that unexpected direction, which means that the top contributors are more likely driving these unexpected associations. Okay, so to wrap up here, we found that the community-level functional shifts are often quite different from the species-level expectations. And there are 2 possibilities on why we observe these. First, the selection force might be slightly different for individuals within a species and species in a community. And second is that maybe those species-level species-level changes we have seen aren't adaptive to warming climate after all, which seem to be a bit more controversial, but there actually are some more recent studies supporting this idea. And indeed, if you look at the species-level evidence, there are still a lack of consensus. And we also see that there's an overwhelming importance of a few top contributors in driving these functional shifts, which joins the evidence from other systems such as forests and grasslands in that a small number of species can often have very large impact in a community composition. And in our case, the top contributors are also affected by the human-induced land use changes, which means that these changes might be at least as important as climate change in driving the community composition of North American birds. Okay, so now that we have looked at the changes in community composition, it is time for us to take a step further and talk about changes in species interactions. And for that, I will use an example by asking how spatial range shifts might affect the potentials of the predator-prey interactions among North American birds. And this is, again, a collaboration with Jia Xie, my friend and colleague since grad school, and Phoebe Zarneski, both at Michigan State. So answering this question, again, will require many pieces of data. First, we need these abundance and functional trait data, which we already covered in the previous one. We also need a database for the predator-prey interactions among birds. And with these data, we can calculate the interaction potential for each local community over 50 years. And then we can, again, associate these interaction potentials with environmental data I have covered before. Now, let's talk about this data on the predator-prey interactions. This is a database collected from the field observations and experiments. And after filtering, we use a subset of 14 predator species, 251 prey species, and an overall 554 unique predator-prey interactions. But there's a problem. Recorded interactions like these are never complete. So here's an example from the Serengeti protected area in Tanzania, which is probably one of the most monitored ecosystems in the world. So if you assume all the species in that area are interacting with each other, which is what panel A shows, you might get something like 25,000 potential interactions. And if you keep narrowing down by removing those ecologically impossible interactions, such as herbivores eating herbivores, you might get something like 8,000 possible interactions, which is what panel D shows. But how many did we actually observe after decades of intensive effort? 592. So in other words, we see that many ecologically possible interactions aren't recorded simply because of logistical constraints in the field. So what we did here was to predict these ecologically possible but unrecorded interactions in our own data set. So to start, we first compiled a bunch of functional traits that are important for the predator-prey interactions, and they include morphological, ecological, and life history traits. Now, why are those important? Or, taking a step back, why are we using those functional traits in the first place? So let's consider this pair of recorded interactions, the Cooper's hawk eating European starling. Now, we have two species that we would like to predict which one might be a more likely prey of Cooper's hawk. So which one do you think it is? Is it this broad-spectred grosbeak, tiny, cute, colorful, or this majestic bald eagle? Right? I think it's pretty clear that in this case, the one that is more functionally similar to a recorded prey is going to be a more likely prey of the Cooper's hawk. And following this logic, we are predicting the potential prey by their functional similarities with the recorded prey. So first, we constructed this functional space from all of those functional traits that we have mentioned for all the preys for one single predator. And then we compare each recorded prey with all the other species in our data set in this functional space. So if the two species are functionally very different, then their vectors in the functional space will look like this, and it will have a large angle in between. And we calculate the functional similarity by taking the cosine of that angle, which we call the cosine similarity. And we, on the other hand, if the two species are quite similar to each other, then their angle will be smaller, and the cosine similarity, or the cosine of that angle, will be closer to 1. We can then use the similarity to calculate the interaction potential between the two species, the predator and the prey. So their potential will be higher if they're more functionally compatible or they're locally more abundant. So if a prey is recorded for a predator, then we define their cosine or the functional compatibility to be 1. If a prey is not recorded for a predator, then then their interaction potential will depend on how functionally similar it is to a recorded prey, which is this cosine similarity metric. So let's go back to this example of Cooper's hawk and European starling. And remember that we have all their abundance over time across all their spatial ranges. So we can calculate the interaction potential in all grids where they spatially overlap. And we can then look at how the interaction potentials change across both space and time like this. So here, these highlighted communities are where the two species are spatially overlapping, and the shades indicate the temporal trends of the interaction potentials, with red meaning that they're increasing and blue meaning that they're decreasing. And looking at this map, you can clearly see that the majority of places where they're overlapping have been increasing, in terms of their interaction potentials, especially those places in southern Texas. But also you see some places with decreasing interaction potentials, such as north or higher latitudes in Canada and also Arizona. And we can also take a look at another pair that hasn't been recorded in the data set, the rose-breasted grosbeak and the Cooper's hawk. Again, we see that the majority of places where they're spatially overlapping, we see an increasing interaction potential over time, except for a few places with higher latitudes and the southern tips of Appalachian. We can then summarize across all the predator-prey pairs in all the local communities to get a map like this. So this is colored by the number, or the net number of interaction with increasing potentials, with red meaning that there are more increasing interactions than the decreasing interactions. And we can see that across the board is a universal increase in predator-prey interaction potentials, with only a few local communities showing there are more decreasing interactions than increasing interactions. And what's even cooler is that we can actually decompose these trends to see if the predator or prey are driving these changes in the interaction potentials. So this is how we do it. We first plot the temporal trend of predator abundance on the x-axis and the temporal trend of prey abundance on the y-axis. And the shades represent the increasing versus decreasing interaction potential over time, with this reference line in between means that there's no changes in the interaction potentials. Now, plotting all those pairs in all the local communities, we can see the majority of them are lying above that line, which means that they're all increasing over time, which is exactly what you would expect from the map on the left. But when we're looking at the temporal trend of predator abundance, you see the majority of them are more positive, or they are larger than 0. But when we look at the temporal trend of the prey abundance, they're mostly around 0. So this means that the universal increases in the predator-prey interaction potentials are mostly caused by the predators. We then want to consider the local community as a whole and ask what environmental factors have been driving these changes in the species interactions. So to do that, we calculate the species turnover or interaction turnover of local interaction networks. So for example, this is a network with 2 predators and 3 prey species. And here are their interaction potentials in 1970, where like the darker shade, which just means that they're higher interaction potentials. And in 2021, things might have changed to this. And we can calculate this single turnover value between 1970 and 2021. And then we fit a linear model of these turnover values with the changes in climate and land use. So let's see how these changes in environmental variables are associating with the species interaction turnover. So the y-axis here is the association, and positive means that the more changes in environment lead to more changes in interactions. And we first see the difference in the temperature and precipitation and their effects on these turnovers. And to our surprise, they are not significantly associated with the changes in species interactions. But when we look at these human-induced land use change variables, specifically settlements, which include both the urban and suburban areas, and wildlands, we see the changes of these land use types are positively associated with the changes in species interactions. So in this case, the turnover of the predator-prey interactions are actually more associated with land use change than climate change. Oh, sorry, I forgot about that. Which is a result not entirely surprising given how humans have been playing huge roles in the increase of the predator abundance over time. So remember when I first mentioned that we lost about 3 billion birds in the past half century? That study pointed out that the predators are actually among one of the few groups that have been increasing over the past 50 years. And that is mostly thinking to the targeted conservation policies, such as banning DDT or banning lead in bullets, which have really caused a rebound of many previously endangered species, such as bald eagle and peregrine falcon. And on the other hand, we also see some species such as peregrine falcon and Cooper's hawk becoming increasingly adapted to urban environments, feeding on birds, smaller birds in the urban areas, and backyard feeders and rock pigeons. So these might have caused a positive association between turnover and changes in the urban area we just observed. So to wrap up here, we have seen overall increase of predator-prey interaction potentials driven by the increase of predator abundance. And specifically from previous studies and our own evidence, we see a lot of signature of human activities behind these increases. And among the environmental factors we have considered, human-induced land use change are again playing huge roles and sometimes even bigger than climate change in terms of these species interaction turnovers. And I would like to highlight that this is the first large-scale quantification of species interactions under global change. And it marks a great step forward, moving beyond only analyzing the presence or absence of species interactions. But we still need more investigations into the ecological consequences of these changes. Okay, so far I have introduced these 2 aspects of community dynamics under global change, but we are still facing a challenge, and that is the challenge of data limitation. So to have better understanding of how global change might affect species interactions, we really need to fill in these missing interactions that are ecologically possible. But a big question is always how. In the previous project, we used one method, which is to predict interaction based on existing records and functional trait data. And the second answer is actually deceptively simple. It's just to collect more data. But you might ask, if data limitation is because we don't have enough people in the field, then how can we get more data without sending more people in the field? So we already have a huge army of collectors around the world, and finally time is ripe to elevate their data to a different level. And here, I meant the extraction of biotic interaction data from citizen science platforms using large language models. And this is a truly interdisciplinary collaborative effort with Tab Hajmadin, a senior computer science student here at UMich, and Xiaohao Yang, who is a PhD student at CEAS. And here, by citizen science, I meant broadly the participation of science by anybody in the general public. But since we're talking about birds, I am zooming into one particular platform, the eBird. Yeah, so eBird is one of the most popular citizen science platforms where people can just upload their observations of birds around the world, really. And it is an extremely rich data source with multiple types of data. So how many of you have actually used eBird before? Yeah, so you are one of those field crews I just mentioned. So here's an example of an eBird checklist that I did back in 2022. And I was there to see one particular bird, the limpet. So these birds are usually found in Central and South America and the southern tip of Florida. But back in 2021, there were some individuals made their way to Houston, Texas, which of course created a huge buzz in the local birding community. So I was there to see them. But after 4 years, surprisingly, they have not only stayed there, but also they kept breeding and have been increasing ever in population. And not only at this site, but also many other sites, really a couple of dozen maybe, like around Texas. So this is very interesting because we're seeing the range expansion of that species happening right under our eyes. But back to data. What do we have? We have date, time, GPS location, very, very basic things. We also have total species observed, their identities, and also their counts. We also have media, and these are like terrible Digital Beam photos I have because I don't bird with camera. But we have options where you can upload the photos, audios, and videos of those observations. And we're focusing on this particular thing, the comments, where you can just put whatever you think are interesting about these observations. And these comments are often packed with ecological information, including species interactions. So here's a comment for evening grosbeak. And despite a typo and this being a bit long, we can see that it's describing how evening grosbeak is feeding together with goldfinch and mourning doves. And we also see comments like this, which describes the mixed flocking between cedar waxwing and Bohemian waxwing, and comments like this, which describes the American crow or mobbing great horned owl. But why have people not look at these comments on a larger scale? So there are two main reasons. As you can see from those examples, they're not very structured or not written in any kind of standardized way, which means that the extraction by human would be very difficult and time consuming. And to make it worse, there are simply way too many comments. So there are about more than 1 billion eBird observations around the world, and I should estimate about millions of them might actually include things like this. But large language models are, they can solve these issues because they're designed to extract information from unstructured text in natural languages. And they can also automatically process very large number of comments. So here's what we do for these comments. We put a comment into the large language model, along with a prompt that contains the instructions and the definition of interactions. And we ask for two pieces of information. First, which species are involved? And second, what is the interaction? And then we manually review their output and fixing things such as typo. And there you have this species interaction record from the from the comment. So to see how well this method works, we did a test with 500 comments that we know already contain bird interactions from Michigan. And we extracted interactions based on an established protocol from Phoebe, my collaborator in Michigan State. And we compared results from manual and large language model extractions. So on the manual side, I really like to thank my Europe undergrad students, Samantha, Olivia, who's here, and Sophia. And they've been really great in working through all these. And on the large language model side, we have the computer science gurus. We have Tab and Soho. OK. So here's the interaction network from the manually labeled comments on one type of interaction, the mixed feeding flock. So you can see that some interactions are recorded more than the other, which means that there are more comments mentioning them than the others. Now here is the comparison between the large language model extracted network. And you can see that there's only one missing interaction, which is already very impressive. Now, looking across all the type of interactions in this 500 comments, we see that it has an 81% accuracy for identifying the comment or interactions, and an even more impressive 96.5 accuracy in identifying the species participating. And then on top of that, they're also extremely fast. It processes about 500 comments in about 3 minutes, where how long it took for you? This is your data, so you should know. Yes, I would assume. I think it was probably more like hours over a few weeks, right? Yeah. So yeah, pretty impressive. What is even more exciting is that this automatic pipeline can be applied beyond interactions among birds. So we're currently collaborating with experts in the field to extract plant pollinator interactions and plant frugivore interactions in many citizen science platforms and data sources. So to wrap up, what can these large-scale interaction data tell us? So first, we can use those to build large-scale, very high-resolution interaction networks across both space and time. We can also have a very deep understanding of species with conservation priorities. So for example, if you want to know what this endangered bird is eating, some comments might be able to help. And it can even lead to discovery of novel interactions that were previously unrecorded. And together, it will fuel the in-depth analysis of species interactions under global change. OK, so let's bring all these together. Through my past research, I've explored on the effect of timing on species interactions. In the two projects I talk about today, I focus on the spatial dimension by showing that the changes in functional diversity are caused by a few hyperdominant species that strongly respond to human-induced land use changes. And also changes in the predator-prey interactions are caused by strong increases in the predator abundance associated with the human-induced land use changes. And finally, these dimensions can be further combined with the big data on species interactions extracted by large language models on both the spatial and temporal scales. Now, obviously, these research can't immediately solve everything, all the problems we face, or the birds face, really. But at least they can shed some light on how to better design conservation strategies. So for example, we have a consistent theme of human-induced land use changes in our results. And land use policies are generally more actionable on local and regional scales. And they can have immediate effects on the fitness and biodiversity of birds. Now, looking forward, and again, this is just like some jargon for job talk, but bear with me. So I wish to extend my work by combining both the spatial and temporal aspects to truly describe how ecological dynamics unfold under global change. And specifically, I want to ask questions like, how do both spatial range shifts and phenological shifts affect species interactions, and how interaction networks are changing over space and time? And I hope to provide some kind of environmental mechanisms in exploring these patterns, asking questions like how interaction strength might change with the environment, and also how we can predict community dynamics with these environmental associations. And finally, I would also like to explore the biological mechanisms behind these patterns, asking questions like how changes in functional composition might affect the potential of species interactions, and also how physiological and evolutionary responses to global change might mediate species interactions. And I will use a wide collection of advanced quantitative methods to answer these questions. So in this talk, I have used and analyzed long-term survey data using hierarchical Bayesian models. And I have also predicted ecologically possible interactions with the novel method, and also introduced a novel data collection pipeline using large language models. And in the future, I wish to use the advanced environment interaction modeling and tools of theoretical ecology, such as dynamic models, to better understand the environmental mechanisms behind these patterns. And I also wish to combine morphological data from museum collections and also data from experiments and observations to better understand the biological mechanisms. Now, going back to the beginning, where I talk about this shining blank screen, and through my research, I hope to shed more light on the dynamics of ecological communities, turning this blank screen into an actual movie that unfolds through both space and time. And I hope my research will provide a holistic understanding for us to better grasp and predict ecological dynamics in the future. So I talk about how the community dynamics can unfold over space and time, but so does my academic journey, and I like to acknowledge the important people along the way. So after 18 years in Beijing, I moved to Chicago, where I first met my undergrad advisors, Kathy Pfister and Alison Anastasio. And then I moved on to Rice University in Houston, working with Volker Rudolph for my PhD, and also received a lot of help from my two community members, Lydia Boudreau and Tom Mitter. And right now, I'm working here at UMich with Brian and Kai. And all these people have profoundly shaped my career as a scientist, and I cannot be more thankful for them. And I also like to thank all the collaborators at UMich that I have been working with, and also members of Weeks Lab, True Lab, Postdoc at IGCB, Midas, and all the collaborators I've been working along my PhD and postdoc, without whom many of these work would not have been possible. And here are my past and current funding sources. And thank you for coming, and I'll take any questions. Thank you.
Interesting. One thing that I was curious about is like whether you see any other interesting trends if you break things up by trophic, like what groups these birds are feeding on. So like aerial insectivores, like are they getting smaller faster, or any other interesting trend there besides the
Yeah.
our landscape being dominated by geese eating corn?
Yeah, for sure. So one thing I should add is that we didn't consider the functional or the changes in functional trait values over time, which could be more interesting in like answering these questions. But one kind of message we got from some recent study is that these within species functional trait changes might not be extremely, like impactful in terms of the community level patterns we have observed. But I think I agree with you that it would be very interesting to incorporate first that, and second, look into the finer functional groups. I think that would be very interesting. Yeah. We have plenty of time, so.
Very cool talk.
you.
Really interested in some of your methods. I can't wait to read some of the papers. I have seen at least one paper showing some differences in foraging behavior around bird feeders.
Mhm.
And I guess I'm wondering here, thinking about in suburban and urban environments where there are a lot of bird feeders, but not a lot of shelter for small birds, which are attracted
Right.
to those feeders. If there's any way to incorporate that either in your large language models and comments or otherwise, what might incorporating those data look like here? Or if you already have, what are you seeing?
Yeah, so for that, there is actually another citizen science project, which is like great and very useful, called Project Feeder Watch. I'm not sure if you've heard of that, but basically they're just recruiting observers that have backyard feeders to record what's feeding there, and like later some kind of species interactions as well. So I think those can be used for answering these questions, including how what species have been visiting the feeders and how they are changing along a temporal dimension, and also like urban suburban kind of gradient. Yeah, so I think those can be very helpful in terms of framing some questions around the feeders. But yeah, I think they're generally quite interesting phenomenon for urban areas, and it really has changed both the community dynamics and individual fitness really for birds as well. So yeah.
Super cool work.
Thank you.
One thing I was wondering about is thinking about like competitive interactions between like species in the same trophic level. Like so often we need to like try to infer like who's competing with who with
Right.
like niche overlap and morphology. I'm wondering if there are enough eBird comments documenting like antagonistic but not predatory interactions that you'd be able to use the large language model to try to get like species identities of competitors.
Yeah, quite a few. I mean, Olivia can like speak to that as well. But, like, there really is a lot, and it's like more than you think. Yeah, there are obviously like predator-prey interactions that are antagonistic, but you also see like blue jay fighting everything. Like it's a very common pattern. Like we have one of the undergrad was working on a blue jay, but basically just looking through the blue jay comments, and basically just like blue jay being like a jerk and like fighting everything. It's just it happens a lot. Yeah, but I do feel like like sometimes some birds that are more common or more conspicuous get more attention in these citizen science data, which is, what exactly you would expect for a citizen science data set. So, just like there are some kind of cautions on that. Yeah.
Thanks. It's a really wonderful talk, and
you.
thanks. I'm so happy now I can explain why there's so many hawks around the campus.
Yeah.
And my question is about again going back to the feeders and their like, interspecific interaction at this point. So the majority of the comments on the listed on eBird is done by the feeder, like feeder watching, then they do have a category like stationary concept, like people was stationed at a certain time period and see how birds behave each other. So I wonder the first thing is like because the existence of the artificial feeder, do they modify the natural behaviors, or like do they provide like something unnatural interaction between the species? And second is the what's called the large language LMMs. Do they have any ability to distinguish between some comment made on the actual wild and doing some of the feeder interaction? Because like as again, majority of comments on the eBirds are heavily skewed on those like feeders and urban environments. So I wonder, Kyle, is there any way to classify it or categorize in better ways?
Yeah, thank you. So I would say that it's very likely that those feeders are modifying the behavior or interactions, and we can test that with the data we have. And that kind of delves into the second question of whether we can discern them. Well, you can definitely do that by just including the hotspots that are like natural reserve areas or whatever, or you know, like do like a very general kind of sweep kind of filter by selecting those points that are not in suburban areas or urban areas, really. And I think in terms of large language model, it's possible that you can also get some kind of insight, like just like purely training the model to like recognize those feeder versus non-feeder comments. So for example, if you go back to that evening growth speed comment, obviously that was made on a feeder, right? I think it's possible, but it just need more. We haven't really like tried that, and there are some nuance in often in these descriptions, and it's possible, but we're not very confident. So I think it's probably like more conservative if we just do like that kind of filtering based on location rather than based on large language models.
Yeah, nice talk.
Thank you.
I'm struggling with so you started out with the climate change is changing body size, and then you modeled body size on everything, and then you said that explained a lot of things, but you didn't model the change of climate change on body size, right? So there's a lack of continuity in the setup and the actual finding there.
Sorry, but by modeling climate change and body size, it was, well, we didn't do the species level things for sure. We did the community level things, and then we did fit a model. Oh, actually, I have this slide where the model is. So this is what the model looks like. It's a lot. So we have this year effect. We have these bioclimatic variables, which is where the temperature, precipitation, and those seasonalities are playing in. And then we have these human-induced land use change variables, and like we have all these interactive effects, and finally we have this random effect. So yeah, so basically for each community weighted mean for like all those 7 traits we included, we fit the same model for all of them. And when I was presenting them, obviously I was just like kind of cherry picking of it, but also the entire result is a lot of interactions or associations that I don't have time to delve in. Yeah, so I don't know if that answered your question or.
No, sorry, I wasn't clear. So what on the Weeks paper, right? So they show that they're shrinking.
Yeah.
And so that's a key climate driver, and that's what you open with as a key climate driver, which is super cool, right?
Mm-hmm.
And then you modeled the fact that all the body sizes are based on a mean size.
Oh, I see what you're talking about.
Time.
Yeah.
And so like I'm curious about that. There's a lack of continuity in the sort of what climate change is actually doing physiologically with this sort of mean body size across time. And I just want to hear some, because that could be a huge effect, right? Those are big changes in body size. If it's a common phenomenon, then actually what you're forecasting is not accurate.
Right, I see what you're talking about. So by that, you're saying that we should consider the within species changes in body mass when we're like calculating these community means over time, rather than using a fixed value or functional value or trait value for like all of them.
I think you can't not because there's a really good paper that Brian.
Right, exactly.
Right. I mean, it's
it's
a super cool. I mean, it's a super cool approach. I'm totally digging what you're saying, but like, and it's a cool finding. But like, I think especially if body size, mass matters at all in your result, then I can see how and it's a cool that you distilled it down and you showed, okay, these 25 species are changing, and that's important. But I think without that component, it's like it's just kind of hearsay because we know it's a real thing.
Right. So on that, I do have 2 comments that are might not be able to directly answer your question, but might provide a bit more context in all these. The first is that many of those changes in the morphology are not directly associated with climate changes. They're just showing that they have been changing over time. And this is actually one of the critiques from the reviewers that we got on the paper is that, how can we really say that and compare those climate-induced, functional shifts on the community level when there's not even a very good consensus on a species level that these are indeed caused by climate changes. So there's that. And second, there are some recent papers that did some physiological experiments that show that either the increasing temperature might not actually lead to changes in the morphology, or the changes in those morphology might not be like super relevant when we're considering like drastic changes in the relative abundances, or really like how those morphological changes might really affect the functioning and heat dissipation of individual birds. So that's what I meant, and it should have probably expanded a bit, when I was wrapping up, is that there really is still a general lack of consensus despite all those amazing studies showing that there are morphological changes happening over time. But I think in general, people are still debating on like what exactly are causing those morphological changes. So, for example, Charlotte, some of you might know, a PhD student in Brian's lab, they have found using long-term mosquito data and long-term tree swallow data that the tree swallow body size are shrinking because of a lack of food resources, which are in turn associated with climate change. But like, it's not a very direct association. You can get the association from maybe the statistics, but like mechanistically, it could be very complicated. So those are just the two comments that I thought might be more relevant to your comment on that. But obviously, that's not addressing the fact that we didn't put these into account.
Yeah, it's tough, right? I mean, it's a hard thing to do. I think that if they're changing in body size, the mechanism is not that relevant for what you're trying to do. So the mechanism, why they're changing, doesn't really matter for what you're trying to do. The fact that they are changing matters. The comment about the relative abundance is super interesting, right? Because that could be, and if that's stronger, then that's like, okay, but I guess that's where the tension is. That would be interesting to test out. So yeah, that's cool stuff.
Thank you. And to add on that, we did some very interesting kind of explorations, if you will, when we're like doing all these analyses. We log transform those body size values to make them a bit more normally distributed, right? Because like if you think about like those geese and those ring-necked pheasants, they're very big and they always have like very large, like body size, and changing their relative abundance would lead to like super large changes in the community means, right? So we did some kind of log transformation on that, and what we found is that, well, first of all, geese was still there, but it was not a major contributor for the community body mass. But then some smaller songbirds are showing up rather than some larger body species. So what that means is that after log transforming, you're seeing the top contributors being the species that are more, changing more in relative abundance versus the species that are, like changing both because their relative abundance are changing and they're like have very extreme trait values, if you will. So that is kind of another kind of interesting observation when we're doing analysis that eventually didn't make into the paper, but that's just another kind of random thought I had.
Question.
Yeah, please go ahead.
Thanks. So the second story was this, and I think I'm missing something, but like it sounds like, so you basically expanded the possible things that these predators could eat, and then you found an increase in this interaction. But isn't that sort of self-induced? Because if you're expanding their possible potential for interacting with stuff, wouldn't it make sense that they would then have more interactions?
Oh, I see what you're talking about.
Like, sounds circular, but I feel like
yeah, so they're separate. So the potential interactions we found are calculated the same way as the actually recorded interactions. They're both calculated based on the relative abundances and then some kind of functional compatibility metric. And if you look at this figure, and like I should have used a better figure, but this is as far as we can get. There are some differences in the shape of those points. And I know it's super hard to tell, but the triangles are the recorded interactions, and the dots are the predicted interactions. And they are really mingled together, which means that it kind of doesn't matter. And we did like separate them in 2 panels. And like if you look at those distribution of those relative abundances, they don't matter. Like they are all the same. We still see the predators like increasing in abundance, and the preys are basically like having the same abundance slope around 0. So yes, that part ended up not having a huge effect on what we observed, which is cool in its own sense. But we were like kind of expecting some kind of different observations between those recorded and unrecorded interactions. Turned out it didn't really matter. And I should also mention that that data set was, I mentioned it was a subset, and it's also like a smaller data set to begin with. So for example, this interaction was actually observed. Like if you look at the Bird of the World, which is like a more comprehensive encyclopedia that has all these life history of birds, like that actually mentioned that rose-breasted grosbeak would be eaten by the Cooper's hawk, but that was not in the data set that we used, and that's why we predicted it. So that's like another way of saying that first, there are really a lot of missing interactions, even in those highly curated data set. And second, that our prediction does have some credibility in terms of what is possible in nature. Thank you.
All right, we are out of time, but thank you all for attending, and thank you for your talk, Hengxing.
Thanks so much. Birding.